Hello, everyone. Welcome to BioVerse episode number 31, the recap of ASCO and ADA 2026, Asian Biotech on the Rise. Today is my great pleasure to have a lineup of very prestigious speakers and experts in these two fields from these two conferences to join me. In case you are joining this for the first time, my name is Leon Tang. I'm the co-founder of iSuite community in Science We Trust community. I'm also the founder of a BioVerse webinar series. We have been doing this as of today, 3 years in a row now. And also a little bit, start with a little bit disclosure because we have a lot of investment corporate audience in buying for the webinar today. This is really just a discussion educational purpose. Please do not use this for your investment advice. And also this is just our speaker's personal view. Their views do not represent their own company or our platform or the organizers of BioVerse. Probably also just one or two words about BioVerse. BioVerse is really the product of a three non-profit. First and foremost, obvious iSuite community in Science We Trust community, which is a US-based nonprofit focused on Asian biotech. The second one is probably I should go back to. The second one is BioSpark, which is a Boston-based nonprofit, where actually Ethan is one of the co-founders or one of the senior advisor for BioSpark, which have an incoming annual conference. You're gonna see that very soon. And the third one is SAPA, Philadelphia chapter, Sino America Professional Pharmaceutical Professional Association. So three nonprofit, US-based nonprofit together created BioVerse about 3 years ago. Again, go back to the disclosure, and then let's dive into today's speakers. It's really my great pleasure to have all my friends, each one of them is really good friends for many years, to join today's webinar. Before I say any word, I would like to invite them to give a brief introduction of themselves. I'll start with David, which is on the left.
Great, thanks, Leon, and hi everyone. My name is David Dai. I'm the senior biotech analyst at UBS, and one of the 4 analysts on the platform right now. We cover pretty much, 80, close about 100 companies now within our UBS platform. I focus mostly on oncology. I have about 18 names on the coverage, including the VEGF PD-1s, which you're going to hear about today, Summit, BioNTech, and targeted therapies like Nuvalent, which was just acquired yesterday by GSK, as well as other modalities. So so oncology is sort of like my bread and butter. And I was at ASCO and happy to share my sort of recap of what I heard at ASCO and go from there.
Fantastic. By the way, Dave and I were both at Barclays. So they'll tell you we do know each other way back. And so next, Ethan, a few words about yourself.
Yeah, so hi everybody. My name is Ethan Xu. I'm the VP of Bioinformatics at Aspen Neuroscience. And on my spare time, I also work regularly with BioSpark, especially running the BioSpark monthly news, and happy to share my thoughts on the ADA.
Fantastic. So Vilash.
Thanks, Leon. Hi, everybody. I'm Viraj Parekh. I'm Senior Advisor and Vice President in the Oncology Practice at Lumanity. We're a global life science and healthcare consultancy with over 1,500 consultants with soup to nuts kind of support, which goes from all the way from preclinical prioritization all the way to commercialization and post-commercialization value access, peer, medical communication. So again, across the board, across the spectrum, across the life cycle of the drug and platform. So very happy to be on board and be part of the discussion.
Fantastic. So just for the audience, your information, right? So obviously our speaker very experienced. If you want to learn more, their bio and the LinkedIn profile were included in the advertisement. So you can go there. A few quick housekeeping. Obviously, we're going to talk a lot about a lot of data from the publicly traded company and from some of a private company, and there's probably a lot of investor in the audience. So when you ask questions, we really encourage you to do so. When you ask questions, please include your name and your affiliation. We will not take any anonymous questions just for the compliance and the non-compete purpose. So keep that in mind. And also when you ask question, please type it in in the Q&A box. We'll keep looking at that all the time. And our speakers will try to address it lifetime. If we cannot, we're gonna follow up later on. All right, let's dive right into the topics. This is what we're gonna talk about today. 2 bullet points from ASCO, 1 bullet point from ADA, a little bit tidbits from ASCO and ADA myself. And with that being said, I'll first pass the baton to David. David, please take from here.
Great, thanks, Leon. Yeah, so for my topics, I want to kind of focus on 3 key topics of discussion at ASCO. One is the PD-1 VEGF space. The other one is ADC. Another one is other sort of interesting target therapies that are relevant to my coverage, but also in the greater oncology space as a whole. So maybe we can dive into the VEGF PD-1 story first. So maybe a quick background information on some of the things that that we're heading into. ASCO, everybody was looking for this summit ivonescimab plus chemo data in squamous non-small cell lung cancer. This trial is called Harmony 6 study. Basically, it's ivonescimab plus chemotherapy versus teslizumab plus chemotherapy. Teslizumab is a PD-1, so basically it's a VEGF PD-1 combo plus VEGF PD-1 bispecific antibody plus chemo versus PD-1 plus chemo. Very much a head-to-head trial, and if you look at the whole space, there's not many trials that's comparing your treatment versus PD-1 chemotherapy. So it's a pretty high bar to start with. And so, very much a well-designed trial, just looking at ivonescimab versus teslizumab. And, maybe quick background on this one. Last year at ESMO, we actually saw, a PFS readout for this trial where, the company showed a hazard ratio of 0.6 with ivonescimab plus chemo over teslizumab plus chemo. So, you're looking at a 40% risk reduction in terms of, either progression or death, right? So it's pretty impressive data for the Harmony 6 PFS readout. But the question then is, how likely is the PFS going to read out to OS? And there's quite a bit of debate on that. And, quite frankly, we actually hit the brown end of that, that debate because, when we look at Summit, basically since October of last year, there's quite a bit of an activity all the way until maybe early, February when, the sentiment improved. But, basically the stock traded down from $20 to $15 from October to February, and then you can rebound back. It's probably just driven by the fact that nobody really knows what the PFS to OS can translate. And so I would say this is definitely one of the most important data points that people are looking for, whether a PFS benefit you're seeing for our Harmony 6 will translate to OS. And quite frankly, we saw that, right? Heading to that readout, people were expecting a 0.72 hazard ratio. Some people are a little more bullish. They said 0.7, less than 0.7. But most of the people that I was talking to think that a 0.72 is sort of considered a bogey or a good benchmark, where you're going to hit static. And what it showed was a 0.66 hazard ratio, right? This definitively tells you that a PFS benefit, for in a Harmony 6, what we saw at ESMO translate to OS benefit here. So I think this is a very very good data here. When we the top line read out early in the morning for the plenary session, and quite frankly, when we a lot of investors already email me saying that this is a home run scenario. And so I'll probably dive into the more detail for next slide. Basically, one thing that I really like about this curve is, first of all, we see separation, at 6 months, right, on the OS side, whereas PFS, we see separation at 3 months, right? This means that we're actually seeing, a little bit delayed probably because, patients who progressed after, after ivonescimab were probably treated with subsequent therapies like, chemo and IO. But then, after the therapies, you'll see the curve continue to separate. So at 6 months, we're here, on the right side here, and basically after the 6 months period, we see the curve start separating, and we see the separate continued over the course of 24, 27 months, right? And so this separation means that there's quite a bit of OS benefit, right? Even with this initial benefit we're seeing here on PFS side, the benefit still continues, right? And so I think that's an indication that the OS benefit is real. The second thing is what we saw was a kind of a flattening of the treatment curve on the top, where after 21 months, you're actually seeing this little line flattening out, right? So in some ways, this tells you that there might be a long tail benefit that's contributed contributed by the PD-1 arm, right? And which was one of the key debate coming out of this readout, say, hey, with VEGF, affecting the PFS benefit, can you actually get the PD-1 benefit here? And I do feel like just based on this data here, there is a long tail that we're seeing prolonged overall. And then the third thing I just want to mention is, you see this last drop. Sorry, before, so just on beyond 27 months, you see one patient dropped, right? And this essentially affected the MOS readout, right? So now the MOS becomes 27.89 months. But one of the discussant or the PI during the oral presentation said was, if this patient did not drop, then the OS will be unreached, right? So with this patient dropped, you're seeing about 4 months of MOS benefit here. I wouldn't really put too much emphasis on that MOS number. I'm actually more focusing on the on the hazard ratio number, which is basically looking at area under the curve, right, between the treatment versus the control arm. And I feel like this is an indication that this drug really works and it reduces chance of death by 34% overall. So this is a very impressive data. Next slide, please. And so on the contrary, though, when we, during the plenary session, after the initial kind of oral presentation, discussant Dr. Julie Brammer came in, and she actually raised several important points. And I would say a little bit more question. She actually provided a little more critical analysis on this data set, and in some ways, it's it's fair, right? I think some of her criticism is fair, but I also feel like it might have, caused some of the, more uncertainty around that data set. What she said was this benefit only happens for Chinese patients, cannot be translated. I mean, implying that there's going to be limited translation to global, which I do feel like, it's arguable. And then in this interim overall survival, of course, it's interim because it's the interim overall survival. And then one thing that she mentioned was this, a patient does not seem to benefit in the patients who are 65 years and older. So on the right side, you'll see that in the red arrow, you see patients who are 65 years and older, the hazard ratio is only 0.93 versus patients who are under 65 years old, which has pretty impressive hazard ratio of 0.43, right? And as a result, she's thinking that this benefit is not, it's not present for patients who are 65 years and older, right? And so, so that's why she raises the question of whether this could benefit in patients who are, who are younger, right? Or actually, sorry, who are older, right? So let me move to the next slide, and I'll make some argument here. And as an analyst, what we do is we try to put together some additional data points for our investors, but also try to provide some additional views and how to kind of think about this data set. So so I get it. I mean, her point of view is very valid, right? In patients who are 65 years and older, you do see a lower benefit here. But then, if you look at other trials, it's the same ivonescimab, for the Harmony I trial, which is EGFR mutant non-small cell lung cancer. And also, if you look at Harmony 2 trial, which is a ivonescimab monotherapy versus pembro monotherapy in frontline non-small cell lung cancer patients, both squamous and non-squamous population here. And what you see here is that the benefit of this difference in terms of age is diminished. There's no difference, to be honest, right? In fact, in the Harmony I trial, you see, the less than 65 years old patients actually have worse outcomes than the patients who are greater than 65. Harmony 2, the very consistent age hazard ratio between the two age groups. So basically tells you that in other trials outside of Harmony 6, we're seeing ivonescimab benefit translates in other studies, right? So that's why I feel like this Harmony 6 data might not be 100% related to the drug itself. It could just be a trial design. So the next slide. What the company has mentioned was a summit, especially after this oral presentation, was that they went back to look at additional data, and this again from Harmony 6 trials, and there previously there was a PFS data readout, and then when they kind of and reanalyze those PFS numbers, right? What they saw was that there's imbalance of patients with higher target lesions in the elderly group, and also there's more brain mets in the elderly group. So 65 years of age patients have way more imbalance towards basically patients with higher tumor burdens, tumor lesion size, and also with brain mets. And so that's what's offsetting this, this difference here. And so if they, so then what you can see here is that, before, the PFS, was around, PFS hazard ratio 0.88, right, for, patients who are 65 years and older. But then if you remove those, those imbalance, take out those covariants, patients with higher tumor lesions, brain mets, then the hazard ratio actually went down to 0.69, right? And so that's actually quite similar to what we're seeing here for patients who are less than 65 years old and of age. So this tells me that this trial is really just imbalanced, with more patients who are a little bit older, probably a little bit more frail, a little bit has higher tumor burdens overall. So as a result, I think this is a trial specific issue and not a drug specific issue. And because the drug definitely works, and then this is, and we've seen, data across, different subgroups currently, right? And then next slide, and so then if you just extend this to other, the standard of care, which is Pembro, right? So for Pembro studies, we see there are two important studies. One is KEYNOTE-407, which is in squamous non-small cell lung cancer, where Pembro, it's Pembro plus chemo versus chemo in frontline squamous non-small cell lung cancer. And what we see here is that there is some difference between 65 years of age versus patients who are younger. And same thing with KEYNOTE-189. There is some difference there, but not as crazy, right? So this tells you that Pembro, is somewhat affected by age. But when we brought this data to KOLs, we quite frankly, surveyed, the KOL and asked them, hey, what do you think about the patient age? Does that matter in terms of your adoption of Pembro? They said, no, not really. I mean, because patients, there's still prescribed Pembro in patients who are a little bit older. Granted, maybe the patients who are really old, like say 85 or 90, right? These patients who are really frail, they probably use monotherapy instead of a combo. But then, combo approach is especially Pembro plus chemo is used pretty much across the entire, age groups, right? So, to be honest, if you look at this profile, there is difference in terms of age, but then that does not stop them from adopting. So to me, this means that, number one, I do feel like, this drug works. And number two, I think Harmony 6, data is probably, restricted to that data set, especially the, maybe patients who are a little bit older. And number three, even if they show a little bit older, as long as it's not greater than 1, for hazard ratio for patients who are 65 years and older, I think physician will still use ivonescimab in their in their patients, especially since there's no other therapies out there. Quite frankly, one of the decision factors is just based on safety profile, right? When we look at the safety profile, Leon, the safety profile looks pretty good. So if you look at the next slide, you'll see that this is indicating that there's especially when we talk about VEGF-related therapy or VEGF-related AEs, there's quite the profile looks pretty clean, especially looking at hemorrhage. Hemorrhage is one of the key factors for VEGF-based therapies, right? Especially in squamous non-small cell lung cancer patients, where patients who are smokers and they have chance of bleeding. And when we look at this profile of patients who are grade 2 or 3, you're looking at 7% for grade 2 and 2% for grade 3 hemorrhage. So in some ways, this profile of bleeding risks and quite frankly, the safety profile overall is pretty favorable. So I think this will not stop them from stop physician from prescribing this drug overall. Viraj, I know you have a question, so I'm gonna just stop here and just
No,
say.
I had a comment as well, David. So other striking thing for me is that if you look at the hazard ratio for the PDL1 negative population, it's actually lower than the PDL1 positive. Again, um, demonstrating the impact of adding the dual VEGF action to a PD-1, because we all know that the chemotherapy backbone essentially is needed because the PD-1 alone is not enough for the PDL1 negative population. So again, that's a really good sign where you show that adding the VEGF is doing what it needs to do in the population where PD-1 alone is not doing the job. And secondly, I think also if you look at the liver mets patients, again, much better data, higher lower hazard ratio, better efficacy versus no liver mets patients, which is also great because you would expect the VEGF, these are vascular tumors, and you would expect that the dual VEGF action would have an impact, which is then translating into a better, hazard ratio. So essentially, those all of those points add to the fact that yes, we've tried Avastin or other VEGFs and PD-1 separately. They haven't done anything in non-small cell, but doing this in one molecule as a bispecific and adding that VEGF dual action is making an impact in patients that are higher unmet need where PD-1s alone were not doing the job. So I think that's also, I think, really important to kind of bring out with this
Yeah.
data.
Thanks for that, Viraj. And you raised very important points, right? Which is that across different subgroups, we do see pretty consistent hazard ratio, right? And especially we're talking about like patients with liver mets, patients with PD-L1 expressions, right? And we see the pretty consistent hazard ratio benefit across different subgroups. And somehow the discussion was just harping on that age group, right? Being like, hey, older patients don't respond as well, which I do feel like it's she's overreacting and quite frankly being too critical about that, about age being a key factor. It's because this is quite frankly a randomized trial, right? And age is not pre-specified, right? And if you look at other subgroups, you do see consistent benefit across the board. So I do feel like other physicians are also pretty excited about the profile overall. So I do agree on that, Viraj.
David, I had one question that what you all think about would be great. And Leon, too, is that so, of course, this is China only data focusing on squamous. If you compare, of course, Summit is running the Harmony 3 trial, which is both squamous and non squamous. Of course, in the US, the percentages are very different, where most patients are non squamous and a much smaller 15 to 20% are squamous. So, what are you all envisioning? So, I know that they're running Harmony 2, which is, of course, China and non squamous, but are we envisioning that this data will translate in the Harmony 3, which is, of course, against Keytruda, which you showed that, of course, but the non-squamous benchmarks are slightly different, right? I mean, Keytruda, if you look at median OS, non-squamous is, of course, higher versus squamous. Of course, the chemo doublet is different, but just wondering what is the sense, you all have for how that's going to translate in non-squamous and the Western trial with Harmony 3?
Yeah. Okay, great. So I will definitely, tell you my, from analyst perspective, how I think about, the translation to Harmony 3, and by the way, we just published on that too. Basically, I think that it's likely translate from China to global, and there's, I would say one thing is, if you look at all the trials we've seen from China to global, there's quite a bit of translation, right? We've seen data translated from, the Keynote 042, China versus China versus global. It's similar hazard ratio. Trust 1-2 similarly. We also have Flora trial. That's Flora, Flora China versus Flora global. Very very similar hazard ratio. So in some ways that gives me confidence that China trial will translate to global trial overall. And at the same time, for Harmony 3, same thing, right? They're enrolling patients who are across different age groups and subpopulations. I do feel like and the fact that they're more they address that imbalance we're seeing for Harmony 6 means that that issue with higher tumor lesions, brain mets probably not exist. So I do feel like that will be imbalance. That imbalance will go away. For the Harmony 3 trial. So I do feel like that's also good indication that Harmony 3 trial will likely translate overall. And then in terms of squamous to non-squamous population, I think that's definitely a key question that, quite frankly, we think there's a couple of data points to back it up. One is look at the Harmony 2 trial, right? You will see pretty consistent hazard ratio between the squamous and non-squamous population, right? And so that's the indication that the non-squamous and squamous population, especially at least as a monotherapy over pembro monotherapy, works well. Now we don't really know about doublet, right? Maybe doublet is going to increase, but we also think that if you believe the mechanism that VEGF PD-1, bispecific is able to kind of reduce, the counter binding, increase the sort of like provide a better tumor microenvironment for T cell attack, right? All of that, it might be able to, drive up more benefit on the PFS OS side. So I do feel like that's also going to translate. And we saw that for squamous population here. And we're seeing the 34%, relative risk reduction, right? For the Harmony 6 trial, right? This indicates that this mechanism definitely works and works better than a combo, right? PD-1 plus VEGF combo for sure. So I do feel like, if you believe the mechanism of bispecific approach, I think it's likely that you'll see this squamous data translate to non-squamous. All right, so that's just my analyst point of view, and we publish on that too. So let me know if you guys have any additional questions on that too. Great, and so moving to other VEGF PD-1 stories. I want to go quickly on some of those discussions. One is, Summit also provide, some of Akeso actually, they provided data from ivonescimab plus FOLFOX6 in frontline MSS high CRC. And this is a China study. It's a phase 2 China study. And what we want to see was whether Ivo plus FOLFOX6, which is the current standard of care in the Western population, will replicate the data for the China plus FOLFOX and FOLFIRI. By the way, it's a triplet. Right? It's a little bit different, right? But so what we see here is that there's definitely consistency in terms of CRC in terms of OR, right? We're seeing 71% OR. On the right side, you see this graph where we kind of point it out that across two different dose groups, there's 71% OR, and we saw pretty impressive 9 months PFS, which is probably the more important data point, right? This means that when you look at the how it's trending, it's going to be more like 14 months of PFS, and this is better than the current standard of care, which is FOLFOX, FOLFIRI, which generates about 10 to 11 months of PFS. So in some ways, this at least from phase 2 trial shows you that when we add a PD-1 VEGF bispecific on top of FOLFOX, you do see a PFS benefit than the current standard of care. So I think this is a pretty impressive data overall. And on the AE side, we also didn't see a lot of discontinuation. Like discontinuation is like 4%. So in some ways, that provides confidence that the safety profile also looks pretty consistent overall.
David, I was just wondering, in this case, I mean, typically in the Western population for RAS mutant patients, RAS wild type, of course, you're getting EGFR combination, but RAS mutant probably is bevacizumab, VEGF. So with the data on the right, probably the median, the FOLFOX6 or FOLFIRI is again underscoring that adding separate VEGF and PD, just is not as good as probably the PD-1 VEGF combination, which can add more value than just adding an Avastin. So I don't know if they did a RAS. So I'm just guessing this is broader population in MSS, but they do a sub-analyses on a RAS mutant versus RAS wild type, or
I don't recall whether I
Okay,
don't think
okay.
they enroll RAS patients. I think this is just wild type for most part, right? And so I think it's a different set of patients. So I don't think they include those patients here. And so just with respect to RAS, it's probably different therapeutic class they can address, which Raj, we could talk about later. But I do feel like this is a wild type population where they'll show more of greater kind of benefit. And by the way, the current standard of care is beva, right? Bevacizumab plus FOLFOX.
Yeah, that's for RAS mutant,
right,
correct? Yeah,
right.
yeah. For RAS wild type, you got EGFR. Yeah, correct.
Yeah,
Okay.
exactly. So beva, it just doesn't work, right? As well. I mean, it works, but we're looking at 10, 11 months of PFS here, right? But then for this trial, we're seeing 9 months of PFS, at 76%, 70%, 76%, which means that if you just draw a kind of a proportional hazard, curve, you get to about 14 months of PFS in some way. So at least directionally speaking, this tells you this might be a little bit better than bevacizumab plus FOLFOX. Of course, this is a single arm study. We'll probably need a randomized control study to really see the real hazard ratio difference between those two arms. And this is where I think Summit will is running a trial for CRC called the Harmony 3G study, right? So I think that's just where they'll probably more data next year. Hopefully next year or later next year we'll see more data from that. But at least this one tells you, directionally speaking, that VEGF PD-1 it has some benefit in CRC and consistent to their previous data for frontline CRC. Okay, and then next slide, we just want to talk a little bit more about some other PD-1 VEGF in the space. One name that we cover is BioNTech. They have a drug called pumitamig, which is a PD-L1 VEGF bispecific antibody, and they're showing this time around a combo with chemo in frontline non-small cell lung cancer, and this is a global trial. That's important. It's a global trial, because they previously showed some data in the China trial. If you see this, the graph on the table on the right side, they show pretty impressive data from China, right? But we don't really know whether it can translate to to global studies. And this is where kind of come back to your point, Viraj, is that in this data at least we see pretty good translation, right, from China to global. And granted, the smaller populations, like phase 2 trial, but we still see OR translated from 67% to somewhere between 76% unconfirmed, 68% unconfirmed, right, for squamous and non-squamous population. So in some ways, it shows you that this data looks largely consistent to the China study, and this is important because BioNTech is running a phase 3 trial called Rosetta Lung 02 trial in frontline non-small cell lung cancer, and people are wondering whether the China trial will translate to global, and do feel like this data provides that evidence that that global trial could likely become positive. So in some ways de-risks the global study overall, and it also provides an indication that this PD-L1 plus VEGF might work just as well as PD-1 VEGF for ivonescimab. So I do feel like this is a good data point supporting the fact that pumitamig is a potential contender for ivonescimab, at least in non-small cell lung cancer space. Now it's late, right? It's like it's 2 years behind non-small cell. Maybe they'll go after other indications other than non-small cell lung cancer. But I do feel like at least this provides some data that this drug works just as well as ivonescimab. Okay. And then if you look at the next one, we have this data from Pfizer and 3S Bio. The drug's called 4404 Pfizer or SSG J707. It's a monotherapy study in frontline non-small cell lung cancer, a phase 2 trial from China. And quite consistently, this showed pretty impressive data, OR of 67.6%, PFS of 12 months, which looks pretty consistent to the table next slide. Basically, when we compare in the monotherapies or frontline data, across in different ivonescimab, in different VEGF PD-1 indication like ivonescimab, pumitamig, Merck's Lenova trial, we do see that OR looks pretty consistent, and PFS looks largely similar, 12 months. Versus 11, ivonescimab monotherapy had about 11.1 months for frontline non-small cell lung cancer, right? But this is a small study, therefore I think 12.4 months. It looks, basically if you just consider this as higher range of 95% confidence interval, I think it's largely in line to the 11 months. So I think on the efficacy side, on the OR and PFS, both look very similar to other PD-1 VEGF. But one thing I do want to mention is the toxicity seems to be, rather interesting, especially if you look at the TRAEs that's greater than three. Basically, you're seeing a 44%, grade 3 TRAEs as a monotherapy, right? And this is, to me, quite, a little bit concerning, because especially if you add this on top of chemo, what does it look like? And this is worse than, let's say, ivonescimab monotherapy, which is about 18 to 20% grade 3 TRAEs. So it remains to be seen how likely will a combo add on top of Pfizer's drug will be able to like reduce those toxicities profiles. But something just something that I want to flag to all the listeners here, right?
I just want to make one comment, David. I think it's really important because it does look like there's no real differentiation between all these PD-1 VEGFs, right? So the point is going to be, it's going to be a optimized clinical development strategy. Where do you go? How quickly you go there,
Yep.
and potentially what your combination partners are. Because I mean, everybody is of course currently just doing, these with chemotherapy as monotherapy. But I think eventually the plan is going to be how do you go beyond just combining with chemotherapy into either targeted therapy or other IO modulating therapies to make sure that you're clinically differentiated. Like you pointed out, everything looks very similar as of now. So it's going to be very critical across the board for Summit, BioNTech, Merck, Lanova, all these guys, because they're not that far behind. I mean, Summit is a little bit ahead, but all of these can catch up. Especially large pharma who has the wherewithal to launch multiple trials based on initial data that they get from the phase 1/2. So I'm just wondering how this will pan out because it's really interesting space, but I think it's just going to be based on how quickly and how you optimize your clinical development and combination strategy to make sure you're ahead and stay ahead essentially. So
Yeah, you raise a very important point, Viraj, and thanks for bringing that up too. And this is something that we've been thinking about as an analyst, because we do want to know like what will be the next strategy, right, for them to like really win, right? Because we know that it seems like VEGF PD-1 bispecific antibody works, right? Then a combo approach. The question then is where is it going to go from here, right? So this is where I do feel like combo strategies with other ADCs or other IO could be interesting. I mean, I would say like maybe plus ADCs would probably makes more sense because you're looking for a next generation IO plus ADCs, which is next generation chemo, right, to drive deeper response. And this is where things Pfizer, for example, and have started reaching out or have started developing combo strategies with their existing in-house ADCs, like their IP6 program, integrin beta 6, which we'll probably talk about here. And also they have other ADCs that they potentially combine this with. Same thing with Pumitamig. They're also combining with their in-house ADC. So BioNTech has quite a bit of in-house ADC that can work with like the B7-H3 ADCs. They license from Duality. I do think those are good combo strategies. And the important thing, of course, is going to be safety, right? So if you combine them together, will they added toxicity on top? It's things like pneumonitis, ILD. So those are going to be key things to watch. So yeah, we'll wait to see on this, but you raise an important point, and I do feel like in the future it's going to be combo with ADCs. Speaking of ADCs, let's dive into some of the data really quickly. I know we're almost out of time, so I don't want to harping on the data here, but very quickly, SAC-TMT was one of the data presentations that people really care about, and there were a lot of interest for that data set, and what we saw was definitely did not disappoint. We see hazard ratio of 0.35. Just pretty impressive data set, right? Which means that and you're seeing a 12 months PFS rate of 62%, which means that you're tracking just looking at proportional hazard ratio, proportional hazard ratio analysis. You get to about 16 months of PFS, right, overall, and this is significantly better than Keynote 189, which is Pembro plus chemo combo, right? So just for example, for patients who are greater than PD-L1 high, so PD-L1 greater than 50%, the Keynote 189 PFS around 11.3 months, and for PD-L1 low, went to 49%. The Keynote and PFS is about 9.4 months. In both cases, SAC-TMT is better, right? It's tracking very well compared to these data points. So I do feel like this is an important ADCs, Trop-2 based ADCs that's better than Trodelvy, better than Dato-DXd and other ADCs out there. So let's move to the next one, please. And at the same time, the safety looks pretty impressive, right? We saw, I think one of the key things that we just mentioned, Iraj, is ILD and pneumonitis, and we didn't see that here. And so the only thing we saw was anemia, which is kind of expected, right? It's just, target, just basically, but based on the biology of this drug, anemia is kind of expected. But we didn't see anything, more concerning like ILDs, pneumonitis. So I think the safety profile looks very, very good. And then moving on to the next one, which is the Pfizer's IP6 ADC in frontline. Yeah, I would say the data looks okay, but does not look competitive compared to sac-TMT overall. We did see, especially in in frontline non-small cell lung cancer, I feel like everything will be benchmarked to sac-TMT now that, the data looks pretty impressive, tracking to 16 months of PFS. So, so it is definitely competitive, but I mean, it looks good, but it's not competitive. And I would say, the next strategy is probably going to be combining with VEGF PD-1. Hopefully, it can drive deeper response going forward. Another thing to flag is just in the instance of grade 3 ILD is about 7%, which might impact adoption, especially if there's like issues around pneumonitis. So something to flag there, right? Next slide. And
David, are you expecting that some of these ADCs will do a sub-analysis where they look at TROP2 high or IB, 6 higher patients to just see if that data is better? Because I mean, in the end, if they want to combine or they want to differentiate or they want to, carve out a population, because they have the bispecifics, they have the other
yeah.
things, they have, TIGITs. I mean, TIGITs are not doing that well, so I'm going to take that back. But they have LAG-3s and others coming in. So I'm just wondering, is that a potential thing that we're looking at where they'll do a expression analyses to see if the target high expressing patients are doing significantly better than, even if it's a broad TROP2 expressing
Yeah.
non-small cell population.
Yeah, I mean, I think if they're trying to do combo study, maybe that might not be the case, because the thing is, they probably want to start with like, a overall overall population, right? See if the combo works, and then if and then of course they'll do subpopulations, about biomarkers, just see if in either of the subgroups, either PDL, or TROP2 high, low, none, right? Whether there's any kind of response, a differential response, right? Maybe that will be something that they can do later. But I would say, like, to start things off, maybe they just want to, especially when they have a combo, I think they'll probably want to start it with a broad population, and then if the results are a little bit mixed, then they probably want to dive into the specific subgroups and see which one works better. So I think that's probably what they will do going forward.
Yep, makes sense.
Yeah. Great. All right, just wrap up on the ADC side and dive into the target therapies. And so Gilead and Tubulis had a NaPi2b ADC, TUB-040, 040. It's a monotherapy in the platinum-resistant ovarian cancer, phase 1 dose escalation trial, and that looks pretty good. It's a 61% OR, and what I really liked it about is that it's actually deepened from last update at ESMO, where they showed about 50% confirmed OR. So you do see a deepening of response over time, which is encouraging. They also show some some early PFS data, 11 months PFS. So I think that's that's also quite encouraging, especially in a pre-high a highly refractory patient population here. And then more importantly is the safety looks pretty clean with no ocular tox, no pneumonitis or neuropathies, based on some of the early TRAs, TEAs that we've seen here. All right, and then I will just wrap up on a couple of targeted therapies that I thought was interesting, and and then I'll pass on to Viraj to dive into some more stuff very quickly. So Pfizer provided some data from their lorlatinib, which is an ALK inhibitor, in frontline ALK-positive non-small cell lung cancer, and this is a 7-year follow-up, of the phase 3 Crohn study. Last time when they presented data was 2 years ago, where they show 5-year survival data, or PFS data, and that was already very impressive. But then this time around, it's even more impressive, because after 7 years of follow-up, the PFS has not reached. And if you look at the drop from 5-year to 7-year, it's only 5% progression, right? So at 5 years you get about 60% PFS rate, and then 7 years you got 55% PFS rate. So this means you have really durable PFS, and this is quite frankly a very impressive data set. And I think when we talk to the KOLs during that time, people are very excited about this data set. They're saying, hey, this is only PFS too. It's not even OS. The OS will probably be 10 years and beyond, right? So this is very impressive data for lorlatinib. And so then, just very quickly wrap this up. what's happened is the next slide is, we have a company that I cover called Nuvalent, which just got bought out by GSK yesterday. They also developing a drug called neladalkib, ALK positive non-small cell lung cancer. It's ALK inhibitor. It's a fourth generation ALK inhibitor. And the differentiation here is that they have a broad resistant mutation coverage similar to lorlatinib, but then they have much better safety because it avoids TRK. And as a result, patients are able to to stay on longer because better safety profile. And what they showed at ASCO was that they showed pretty impressive OR data, right? You're showing about 46% for patients who are lorlatinib naive, and anybody who has treated with lorlatinib experience is about 26%, 31%. And these are patients who become refractory to many other therapies before. So in this line of therapy, patients do not respond to anything. Pretty much like the only thing that's available is chemotherapy. You get about like 3 or 4 months of PFS, and that's it. But what they show here is more important is the durability, right? They're showing close to about 14 months of PFS in lorlatinib naive, and also they're showing median DOR for patients who responded was not reached, with lower range of 14.5 months, and this is much better than lorlatinib, which was just mentioned previously. And so lorlatinib was showing about 12.5 months, whereas this data is showing a lower range. We're already getting for 14.5 months, right? So you're already seeing much better response, duration response here. So I think this drug, is at least as good as lorlatinib, and the safety profile will definitely be better than lorlatinib. I think this is the reason why GSK, gave them a $10 billion acquisition eventually, and eventually got bought out here. All right, I talk a lot. I'm going to stop here, and maybe I'll just pass on to Viraj.
So David, actually, quick question for you. Obviously, there's another company in the same space called Nuvation, right? So Nuvation is like kind of a fraction of a valuation of Nuvalent. I mean, they both start with N. Some people may confuse them. What's analysts, if you can tell us, like let's say 1 or 2 sentences, why their valuation have such a huge gap?
Yeah, the first thing is Nuvation Bio is only focusing on ROS1 non-small cell, and ROS1 is a really small market, about like 3% non-small cell lung cancer, whereas Nuvalent is focusing both ROS1 and ALK. And ALK is a bigger market, about 10% non-small cell. So basically, the majority of that value for Nuvalent is driven by the ALK inhibitors, right? Basically, if I have to give some numbers around it, I would say 20% of value is probably attributed to ROS1. And the 80% is attributed to ALK, and this is probably one of the big difference. Also, another difference is just the profile of the drug. For Nuvation Bio, they have pretty good survival benefit, but then it's also very toxic. A lot of liver tox. As a result, patients discontinue from it. Therefore, especially if the company's arguing for a stacking going forward, it's going to be tough, because the thing is, patients do not stay on as long, because a lot of patients already discontinued treatment overall. So this is why I think there's a kind of disconnect between the evaluation here, which I do think makes sense.
Right. Thank you. I mean, also as a reminder for people always wondering whether the US biotech are really under a lot of stress. Well, here we go. You have like a 10 billion plus acquisition. Right? As long as you have a good drug, right? Really is kind of let's say it's not even like a makeup or blockbuster, peak sale, right? In my opinion, that's already justified over 10 billion, acquisition cash. Anyway, let's move on. We are really behind the schedule. If you can speed up a little bit, I really appreciate that. I know people probably have other stuff. So Viraj, the floor is yours. Please go ahead. Oh, why is here? Whatever.
Oh, okay. Okay, so we couldn't go without, of course, talking about daraxonrasib. So if you want to just go to, yep, go next. Okay, yep, of course, the star data that was there, the blockbuster, of course, everybody saw it. There's a standing ovation, so there's a black and white kind of silhouette of that. It was absolutely fantastic. And it's not only because I think it was, KRAS has been undruggable for a really long time, and we've been trying to go beyond sotorasib and adagrasib for a while. And of course, RevMed has done that with a lot of their RAS on, especially daraxonrasib. They have a bunch of they have zoldonrasib, G12D, G12V, G12, I think R as well. So basically, they have a very strong pipeline, right? So, and this is of course the first time that we've seen this kind of data with a broken the 12-month median survival barrier in second line plus PDAC by any agent across the board, IO, non-IO, chemotherapy, targeted therapies, which is absolutely unbelievable, right? So you have doubled OS, doubled PFS, and you have the ORR is 3x. So essentially, this is just basically completely paradigm changing, right? So this data almost is comparable to first line data for what you see with Gemabraxine. Historically, of course, modified FOLFIRINOX is probably slightly better, but the tox profile is significantly better in this case, right? So if you look at the tox profile, so I mean, the idea is that of course this is a blockbuster in second line, but of course the idea is we want to take it as early as possible, which RevMed is probably also thinking, and they're probably thinking that we are going into move into first line metastatic PDAC, right? Again, with or without combination with either. There are other G12Cs or G12Ds or others or Cs or potentially with a lower dose chemotherapy or modified FOLFIRINOX at a lower dose or a 3-month dosage. So again, I think it's absolutely fantastic data. It's paradigm changing, and only a couple of things. Of course, this is 8.5-month follow up. So of course historically RevMed has changed their messaging from a pan RAS to a multi-RAS, suggesting that it doesn't hit everything equally. So we don't know if this, G12Cs are doing better than the Vs versus Ds. Yes, the overall population is all covered here. It looks really great, but that would be good to find out. You may have some late onset resistance mechanisms that do prop up. So that's something to cover. I think that's something that RevMed will think about when they're thinking about novel combination strategies and frontline. Is that only with chemo? Do we try to do in-house with their own G12Cs, or do we look outside with, other novel G12Cs? Right? So I think that's something that they really probably are thinking about right now. And I think the other aspect is that this is all G12 population, which is, of course, the majority of PDAC. But if opportunities beyond PDAC and G12, you'll be to better understand how this multi-RAS targeting of daraxonrasib will pan out. And again, this is probably going to be, a optimized combination strategy. And once they, iron out details on, how or what it's hitting, how it's hitting, and what best mutants you can target with daraxonrasib. So, but again, I'm not going to belabor the fact that this was absolutely fantastic and is going to change the paradigm for metastatic PDAC for the majority of the patients, right? So, next slide, please. Okay, so this is just a couple of other therapies focusing on non-small cell because they have a whole bunch of actionable genomic alterations. And then this is a couple. The right, of course, lorlatinib, David already talked about. The left is serpercatinib, which is again showing a different patient population, significant delay in disease recurrence in RET fusion, which is not a big population, but still the data is absolutely fantastic. Again, so if it's for stage 2 to 3A disease, I mean, actually stage 1 to 3A, 1B, 3A, it showed a hazard ratio of 0.172, basically 83% reduction. If you look at the EFS, it was 2-year EFS was 91%. So again, if you do a little bit of the sub-analyses, the hazard ratio was actually even lower at 0.125, which is 87.5% reduction in risk of recurrence. This is again potentially higher or as high as what we've seen with EGFRs and ALKs in the same population as adjuvant. So again, very, very nice data, very convincing. Again, underscoring the fact that we need to do more NGS testing, not just for metastatic patients, but as early as possible for especially for non-small cell, where now you have 40 to 60% of patients that have actionable genomic alterations, right? So it's really important that this underscores that. And now you have EGFR, ALK, and now RET as well, that is going to be part of that early stage intervention, which is basically going to increase survival significantly and delay progression for all these earlier stage patients. On the right, lorlatinib, again, David already talked about it, but just one point. So thanks, Leon. Is that essentially what this has done is that it's basically almost a functional cure or a functional long-term remission in metastatic disease, which is very, very, rarely seen in patients, even with targeted therapies in non-small cell, even with EGFR, even with others. So the point is that I think one of the main things is here, if you look at total suppression of brain mets, which is 20 to 40% of patients as they for non-small cell as they progress, and it is one of the major causes of mortality as well beyond liver mets. So there's a 94% reduction, basically a hazard ratio 0.06, which basically means it's completely shut down the CNS progression, which is absolutely fantastic. So I think that is key to show here, and the fact that it's reasonably well tolerated. Again, I know that David talked about next gen agents, which can but the fact that lorlatinib is showing this data in early stage ALK is absolutely fantastic. Okay, next please. This is another one of the agents that is early stage, but for KRAS G12C, again, we have so many agents, G12Cs on, G12Cs off, degraders, other conjugates. But this was, I thought was striking because it has really robust monotherapy data. Yes, it was, it was majority was China based, but they did have US and Australia patients. So again, this is across populations, across geographies. Phase 2 data from D3 Bio for their LS-RASib G12C off next-gen inhibitor, which had a monotherapy efficacy across PD-L1 expression again. So all patient populations, 78% again. So, much higher than what adagrasib and sotorasib see in non-small cell, which is around 35-40%. So basically doubling ORR as monotherapy. And if you look at the combination with Keytruda, again, this is across all PD-L1 expression. So even in PD-L1 low and negative patients, they're still seeing 80% plus ORR and a PFS, 12-month PFS rate of 53.7%. So basically, again, this is in first line where Ada and Soto and others are trying to have trials in frontline with Keytruda or with Keytruda plus. They're trying triplets with chemo. But the idea is that even though it's early, it's probably, I think, 18 or 20 patients, but very striking for me that they've doubled what we saw with with the adagrasib and sotorasib. And again underscoring the fact that you have to do the full genome sequencing as early as possible for all non-small cell patients to make sure that you catch all these G12Cs and ALKs and RETs and NTRKs and others. So, yep, just next. Thank you. I'm not going to dive too much. There was, a bunch of late stage other clinically relevant trials across. So there's FGFR2 for pemigatinib, again, hazard ratio of 0.58 in first line. Cholangiocarcinoma, again, pretty significant. Phase 3 still developing, but really promising in first line for the FGFR2 fusion patients, which is a reasonable amount in cholangiocarcinoma. Exon 20, of course, you got AMI and lazertinib, but again, that tox is really something for amivantamab because of the dual targeting. So this is another phase 3. Again, this is China, mostly China data, but again, first line monotherapy, very strong ORR for this agent in the exon 20, which traditionally, of course, is not targeted by osimertinib. This was a PARP data by Pfizer in HRR, of course, HRD patients in CSPC. So again, we know that PARPs are present in BRCA mutant and HRD to some extent, CRPC, but this is taking to the earlier levels. The point is that this is basically data to support that we can take PARPs into earlier line disease, earlier stage disease, which is basically going to delay recurrence and potentially significantly increase the delay for patients to get to CRPC, right? So this is potentially probably for me a paradigm changing trial for PARPs in prostate cancer. A little bit about the SERDs and the SERDs, of course. Mixed data for this year's ASCO for the SERDs and the SERMs in ER positive BC, but I think there's some silver lining with giredestrant, the Roche readout. So again, it did look reasonably promising. Again, it had some populations that did not reach, statistical significance. But overall, of course, that the field is moving ahead, and you're going to see more and more surge in earlier line ER positive BC. And the last one is basically apalutamide, which is again in perioperative setting, which again showed significant improvement. But again, the data is early for the phase 3, but the idea is again taking apalutamide earlier in the perioperative setting. Next, please. Again, this is the early one. So again, of course, this is with take that with a pinch of salt, because the translation rates are not, of course, as everybody knows, not as high. But again, across HER2, V1s, and the EGFR, MAP kinase targeting across the board, showing some data in lung, solid tumors, data in a post-checkpoint setting. All of these are high unmet need settings. So again, looking forward to how this data progresses. So again, I would like to comment that V1 was, I think, the last session at ASCO, and again they pointed out that it is. So I know that historically it had its baggage, but I think they did point out that W Farms V1 a PK-MYT1 combination had shown some really differentiated data, along with a very favorable safety profile. So again, I think V1 is probably making some kind of comeback, and again, it has commercial applicability across various tumors. So, yeah. Next, please. Again, so I'm gonna go a little bit quicker, but again, this is a focus on IO. Of course, a lot of IO read out, but this is on CAR Ts. Early data from novel target-based CAR Ts made a huge impact at this year's ASCO, especially in solid tumors. So on the left, we have RSL's data, which is GPC3 HCC, which is expressed in about 70 to 80% of HCC patients. This was in relapsed refractory population. The CAR T showed a 66% response rate at RP2D, and a DCR at really 90%. Median OS 21 months. So basically, this is better ORR than first-line atezo-bev, which is at 32%, and a better OS than what atezo-bev has in frontline. So again, of course, this is CAR T, so there are of course other hurdles, logistical. From a safety perspective, CRS was mostly grade 1-2, no ICANS, and potentially so it does look like the safety is better. The fact that GPC3 is a differentiated target seems to be working. So I think it's, and again, these are all patients which are late line. So these are all immunocompromised, beat up patients which have already got dirty TKIs like sorafenib and lenvatinib. So again, the key take home for me is that this was really good response rate for a CAR T in solid tumor for me, especially in really difficult to treat late line HCC patients. On the right, we have Legend Biotech's, again, DLL3, well-validated target, of course, with the bispecific in SCLC. But for the CAR T, again, phase 1 data, for me, again, not, it is impressive. It is comparable to what, tarlatamab shows of, a 30% response rate. PF duration of response is about 6.5 to 7 months. But I think there is potentially value to this, because again, it's a one and done treatment, which is probably much more different from a tarlatamab, which is then associated with longer term use and longer term toxicities. Also, I think the main idea here would be to follow the data to make sure that the CAR T approach can then provide that superior survival benefit and durability, which probably BiTE and other DLL3 targeted agents like ADCs probably will not be able to provide, because that is going to be key to then offset some of the higher costs and manufacturing demands and the wait for to get it. So I think there's value to this. I think there is initially proof of mechanism, there is proof of concept, but I think we need to wait for mature data to see how clinically relevant and commercially viable this is going to be, especially in small cell lung cancer. This is from Colonia. This is the in vivo CAR T, made again a bit of a splash in relapsed refractory multiple myeloma. Again, we all know that multiple myeloma now has 8 to 10 lines of therapy, but as you go beyond 5th, 6th line, again the response rates go significantly lower. The unmet need is much higher. So I'm not going to delve too deep, but again, 18 patients only, heavily pretreated, all fourth line to sixth line patients. All of these patients were triple class exposed, so essentially got proteasome, got the IMiDs, have got the CD38 antibodies, and a majority possess high risk cytogenetics, right? So again, if you look at the data, 100% response rate across dose escalation, 100% MRD negativity again at day 30. So again, if this can translate into durable, responses, which will continue beyond 9-10 months, I think it will be fantastic. From the safety perspective, CRS was mild, transient. Neurotox was manageable, minimal. They're calling it minimal, but I think it's reasonable but manageable. And only a couple of cases of ICANS, right? So again, not as toxic as you would think associated with a CAR T. In vivo, of course, is making is one of the hot kind of spaces right now, and I think this was something that will probably be looked at and followed for some time. Because I mean, if you look at Carvykti, which is of course is potent in late line multiple myeloma. It is of course has manufacturing backlogs. You need to wait 4 to 6 weeks. High cytopenias. So this could be potentially a scalable, off-the-shelf alternative that could be given, even in outpatient clinic. So you're not waiting for the patient to be in the hospital, waiting to see if the CRS needs to be managed, especially if it's grade 3 plus. So again, and the other aspect is bispecifics as well, because they're all in this space, and they do trigger T cell exhaustion, other infectious vulnerabilities. But in this case, this could offer a one and done kind of profile, which is not as toxic as typical CAR Ts are, and much more targeted with the in vivo aspect. Thanks. Just a kind of high level on focus on IO. So, tarlatamab, of course, got its approval earlier this year. So just the, much more mature data that came out. Same for the Majestic 7 trial by Janssen in first line, for the first line triplet in multiple myeloma. Both these, again, have got their approvals earlier this year and are now building on, of course, their earlier approvals in later lines. So I'm not going to dwell too much, but again, this was great data to show that it is still working, and the mature data still looks like what the earlier phase 3 data looked like. Next, please. Okay, great. Thanks. Just a couple of other readouts before we pass the baton to Ethan and go beyond oncology is RP2 Replimune, which is the next gen after the RP1, which is, of course, the OV, which is also then anti-CTLA. So again, this had 36% response rate in PD-1 refractory, and again successfully triggered deep local. So this is of course a locally administered agent which is showing abscopal effects, which is very interesting. I know that IT drugs have their baggage, but I think there's a bunch of companies out there, many of them who are our clients are working really hard, and I think IT will evolve and is evolving at a very rapid pace to again make its place in the oncology landscape. So again, this is one of them. It locally expresses, of course, anti-CTLA4. So basically, then circumvents what you see with systemic CTLA4s, like you see with IPI. And then potentially, it is all in PD-1 refractory. So the ability to then, a proof of concept to then turning immunologically cold tumors to hot with using a multi-mechanistic viral vector. The next one is Obsidian, which is the next gen IL-15 engineered TIL therapy. Again, this is going, which is again melanoma, like RP2 was melanoma data, is a 63% response rate and a DCR of 93. Again, relapse refractory melanoma. Typically, if you go beyond the BRAF therapies and Ipi-Nivo, even the LAG-3s, of course, are looking at a very low response rate with chemotherapy. So this again, does eliminate the high dose IL-2s you need with other, TIL therapies like conventional Iovance and others. So again, could kind of circumvent some of the logistical issues you see with Iovance's drug and the commercial adoption hurdles, again, due to, the ICU-based infrastructure that you need to maintain with Iovance's potential 97% grade 3 adverse events. So again, if you can move a TIL therapy in melanoma to a safer outpatient as an outpatient modality, I think that could really be significant for this agent and such kind of approaches. I am going to stop here. I know it was really quick, but I wanted to give time to Ethan and then Leon as well to finish. Thanks, guys.
Thank you very much,
Yeah.
David and Viraj. So now let's pass the baton. By the way, this is our first time cover ADA. Thank you very much, Ethan. You're first one to help us unpack. It sounds like a lot of data released from ADA. So floor is yours, Ethan. Please go ahead.
Yeah. Yeah, so I will be very quick. Try to be finished in 20 minutes, maybe even 15 minutes. So I only have time to cover three highlights. So mainly Eli Lilly's triple G, which is very much in the news headline. Why I will explain briefly why the glucagon receptor agonism can help. Then I'm going to talk about Pfizer's long-acting GLP-1 and monthly dosing. I will wrap up with one of the hot topic in muscle sparing in terms of muscle sparing weight loss, I will choose Regeneron and showing why they struggle. So to the next, I'm going to give quickly overview why is triple G. So you already have the double G, which is the tirzepatide, which is doing quite well. But as you remember, GLP-1 and the GIP are both GI hormone, basically suppressing the appetite, reducing food intake. The third G, glucagon, although initially a little bit intuitive because it may raise blood sugar, but actually if you pay attention to glucagon, not only they're homologous to GIP and GLP-1, they can actually kind of stimulate the energy expenditure. So you burn more fat. So that's the key thing. That's why the glucagon is one of the hot topics. Now the people are even considering the other double G, the GLP-1 and the glucagon double receptor agonism. That's promising because GLP-1 suppress appetite and the glucagon receptor agonism will stimulate energy expenditure. That's why the triple G really catching the headline because the third G turned out to be very interesting. Then in terms of engineering, you can imagine that glucagon has homology, but it's not just taking the consensus sequence. So when Eli Lilly scientists to engineer this triple agonist, they essentially eventually choosing the main kind of design focus on glucagon receptor, GCCR, so that you chose those peptide sequence. Then you optimize this empirical screening, try to bring in additional GLP-1 and GIP receptor agonism. So that's why it's not easy kind of engineering. So I'm only going to talk about obesity part of the trial. There's another basket called sleep apnea, and there's another basket called osteoarthritis, which is probably minor. I will only put in summary. Go to next. Then in terms of the design, each dose, other than the placebo, having those loading dose. So the 4 milligram, you have the 2 milligram as loading dose, then 9 milligram, 12 milligram. It's kind of try to show the dose dependent. Of course, some subset of patients will go through the extension for open label, but I'm going to go to detail. The primary endpoint for this part of obesity is measured at 80 weeks. You try to see the what's the weight reduction. Go to next. And you can see the number is quite impressive, right? It's in basically, I don't have time to go through what is the efficacy estimate versus treatment regimen estimate. Essentially, efficacy estimate will just consider all the patients, even they drop out. Then you get a slightly better number, typically than the treatment policy estimate. But you can see that it's dose dependent. So from 4 milligram is then getting better to 9 milligram, then 12 milligram is the best. Can reach as much as 30% weight reduction. That's unheard of. So on average, it's like 28.3% on the highest dose, which is very impressive. So that's why probably still a lot of headlines, and that means it will probably putting tirzepatide into a very interesting point. Lilly is probably their product is competing against itself. So go to next, we will see the GI, no, there's other secondary endpoint. It's also very interesting. So the second endpoint, because you try to the glucagon receptor agonism will stimulate energy expenditure, you definitely expect to be better, doing better for the lipid profile. That's why the triglyceride lowering is also better in 9 milligram and 12 milligram. LDL cholesterol also lower than the blood pressure. You can see lower the blood pressure. Then inflammation biomarker like high sensitivity CRP, you also see improvement. So overall, and even the kind of glucose, because for pre-diabetic patient, quite a number percentage of the patients on triple G eventually going back to the normal glycemia. So that's overall a very impressive data. Go to next. We will talk about now what about the safety, right? But the GI tolerability issue won't disappear because any of the 3G will have some kind of GI tolerability, but it does not getting too much worse. If you look at the first, like at least one adverse event in the treatment, you will say placebo is 80%, 4 milligram is 87%, 9 milligram is 89%. So not that much significantly worse than the placebo. So I think that's quite acceptable. Then serious AE also quite similar. Placebo is 5.5%, and those three groups either 7.7% and 10.5%. So overall, I think the safety profile is also very acceptable. Yeah, go to the summary, and you can see in the summary that not only it improved the weight loss number over tirzepatide, now some patients even losing 70 pounds. That's really impressive at 104 weeks. And then the FDA lowest threshold is 5%. Not only, that's easily all patients meet that lowest bar. Then 85% lose at least 15%. Then you're improving the knee osteoarthritis. You also improve the sleep apnea, lower the lipid panel. Overall, I think this definitely will be a newbie on the market, will be kind of very much patient will welcome this addition. So now let's move to the next topic, which is Pfizer's long-acting GLP-1. And so the reason is Metsera, right? Pfizer and Novo Nordisk bid against each other, try to buy Metsera. And Metsera is a company focusing on how do you extend further the half-life of the GLP-1 receptor agonist, and which turned out to be a potential first-in-class for monthly injection. Go to the next slide. I will give you brief history about Metsera. So Metsera bought another British company co-founded by SRB. The name is Stephen R. Bloom. People remember Dr. Knudsen at Novo Nordisk was inspired by the 1996 Nature paper published by Bloom Lab. So what that lab doing is injecting directly the GLP-1 7 to 36 into the mouse brain, and they suppress the appetite. That inspired Dr. Knudsen to launch the long journey about making the GLP-1 receptor agonist a drug for obesity, not just for diabetes, type 2 diabetes. And you can see Dr. Knudsen's semaglutide and essentially changing the second amino acid to AIB. It's the amino isobutyric acid, which definitely helps. And also, Dr. Knudsen put the lysine residue and putting additional lipidation to increase the albumin binding and extend the half-life. But Bloom Lab, founding the company acquired by Metsera, doing a little bit more. So they think not only they take Dr. Knudsen's modification, they blocking the lysine in the middle, they change it to arginine. Then they extend the C-terminal sequence, eventually putting the terminal lysine residue for lipidation. But not only that, they have some undisclosed secret that they can change in the lipidation at the C-terminus. And we still don't know how it can achieve more than 15 days, but that's their secret recipe. So it's essentially the peptide, even without release by albumin, can bind to the receptor GLP-1. So that's why it's very impressive half-life. Go to the next. I will just showing compared to other kind of product on the market in terms of the half-life. You can see the liraglutide 13 hours. Knudsen's major breakthrough is semaglutide getting to 1 week. Then for this particular compound, and it's called MET-097, which is now called berobenatide or Bero, it's reaching almost more than 16 days sometimes. So that's quite impressive. Even maritide is not a peptide. It's a GLP-1 agonist and GIP receptor antagonist getting similar. But as we know, maritide probably won't be able to compete with the Bero compound. Go to next. I'm going to show quickly about the efficacy. And you can see that even weekly approved therapy like semaglutide at 2.4 milligram per week and the tirzepatide 10 milligram per week, because tirzepatide is hitting 2 receptors. You need to increase those. That's why it's not surprising. But you can see they on average having either reduction of 9% at week 28 or 12.5%, whereas the barrel, even at the monthly injection, is comparable. Although I have to caution, this is not a head-to-head trial. We have to be running a head-to-head trial to venture. But even based on those cross-trial comparison, you can see the monthly injection of this GLP-1 receptor agonist looks quite comparable to those approved therapy. So I'm going to next. Now, what about the?
So Ethan, quick question for you. So this is still long-acting mono agonist, right? They're not a dual agonist.
Not dual agonist, just the GLP-1 receptor. They will eventually combine with the amylin, which also comes metaxera, but that's coming next. But this is already quite impressive phase 2b result. I think they can directly for registration purpose without doing a phase 3. That's my kind of guess. Yeah, so go to the GI. Many people say, oh, it's really hoping that by doing monthly rather than weekly, not only it make it more convenient, you are hoping it can be more tolerable GI. Overall, yeah, that's pretty much, you can look at arm 3 and arm 4. That's the monthly dosing compared to the loading weekly dosing and the monthly dosing. Yeah, overall, the GI profile, it looks quite similar, and probably say not too many dropouts compared to the double G cases. So that's why I would say this, safety profile also looks pretty good. So going to the next, I'm going to just talk about why the market will welcome a monthly injection. Pretty much a cost of good analysis. That's why Wall Street analysts like to do the COGS analysis. You can think about this as auto injector. That's why the reason auto injector is so popular. The PCSK9 Amgen's antibody is still on the market without threatened by too much by the siRNA because it's an auto injector even every 2 weeks. Whereas the siRNA by Alnylam, even 6 months, you need to go to doctor office to do the injection. Even subcutaneous, auto injector is highly welcome. So for the auto injector, if you only do monthly, you only need 13 devices a year. Then MariTide is on that range, but MariTide you have a lot of dosing, a lot of milligrams of drugs injected into the body. But tirzepatide and semaglutide each are weekly. You need 52 auto injector a year. That's a lot of. That's not cheap. So to making those auto injector, you have to store in the refrigerator. Then tirzepatide also you have a lot of higher dose in terms of API because semaglutide typically is go low dose. That's why monthly injection is highly welcomed by the market and definitely would be very competitive, and also auto injector and the lower volume. So that's why
Ethan,
I'm going to.
Quick question for you. So this actually is something new to me. I was not aware. Do you know the percentage of COGS, COGS breakdown between the ingredient and the device for the manufacturing?
It's mainly including device. So that's
I know.
why.
Let's say if you spend like your COGS is $100, how much actually contributed by the device, the auto injector, by the peptide?
That's a good question. I'm not an expert. You may need to talk to some CNC people, and we can have another discussion. Yeah, but definitely they do some modeling. Overall, they're very optimistic, bullish on the COGS advantage. Yeah, Pfizer may have some other detail. So let's move on to the final topic on muscle preservation. The reason Regeneron, if you hear George Yancopoulos keep talking, we acknowledge Lilly and Novo's success, but they are also losing muscle. So many years ago at Johns Hopkins, Seijing Li's lab discovered myostatin, and it's a negative regulator of muscle growth. It's in the TGF-beta superfamily. Looks very promising because knockout mice is like so muscular. They put on the Nature cover because it's like it's a huge muscle mass. So this family, interestingly, are going through two types of receptor. The type 2 receptor is the main binder of the ligand, either the myostatin, also known as GDF8, activin A. They bind to the same set of activin receptor, type 2 receptor A and B. That's the heterotetramer. And now heterodimer, then they recruit type 1 receptor like ALK4 and 5 for downstream SMAD signaling. So because it's a negative regulator, it's really nice. You just want to be a myostatin inhibitor or a activin A inhibitor. And interestingly, bimagrumab by Novartis, now in Eli Lilly, is hitting the receptor 2A and 2B. Whereas Regeneron, their peptide, their antibody is targeting the ligand. They have anti-myostatin ligand antibody, they have activin A ligand antibody. The activin A ligand antibody is for rare disease called FOP, which I don't want to detail. So Regeneron want to see we have two antibody. Let's do all the combo. So go to the next slide. We will design a very complex trial. So why? Why they want to design a trial? Because they believe GLP-1 will leading to muscle loss if you adding on a myostatin antibody, blocking antibody, can preserve the mass, or you can add a activin A antibody. But unfortunately, activin A antibody having some side effect, nose bleeding, epistaxis. For a rare disease with no treatment, FOP, that's acceptable. But for obesity, definitely not acceptable. That's why they're only doing the activin A antibody for proof of concept. But their main focus is the myostatin receptor antibody. Going to the next, you will see it's a compressed design. So basically, because their activin A is unfortunately antibody is IV, their anti-myostatin is subcutaneous. Then semaglutide is also subcutaneous. Then you have to have different kind of placebo for the semaglutide only group. You have to have a subcutaneous placebo and an IV placebo, which is not easy to run this kind of trial. Then the other arm is two arms is trying two different doses of myostatin, 200 and 400, and without the activin A. The third arm is very complex. You combine all three drugs to see, but you do know that the activin A won't be eventually seeking approval for obesity because of nose bleeding and other issues. So that's why the trial is so complex. Then they use the DEX scan, dual X-ray kind of scan to measure the lean mass versus the fat mass loss. So going to next, give you some quick results about this phase 2 trial. Yeah, the disappointing side is that in January 2025, people know that FDA has a draft guidance saying for any anti-obesity therapy, you need to reach this 5% weight loss for clinical meaningful. But you know that you're preserving mass. Sometimes you're preserving lean mass. Sometimes muscle may grow a little bit. Then you have the fat loss. If you have those confounded, then sometimes does not always translate into the total body weight loss as you desire. That's why and Regeneron is in a very tricky spot. They definitely show a lot of good data. So the lean mass definitely increased, the preserving the muscle, but unfortunately, you can see the right three arms, semaglutide plus two different doses of the myostatin, just around 3% to less than 5% body weight loss. Then the triple combination, yeah, it's even worse. It's a 1.9%. But regardless of the baseline low lean mass or not, but this is a very tricky result. What do they do next? Because they didn't reach the 5% threshold, they have to negotiate with FDA agency to see what to do next for phase 3. So it's a very tricky situation because you have preserving the mass, muscle mass, but you don't lose the fat enough. So that's why it's a very mixed message at the whole ADA conference. And Regeneron people can tell you that they're not to know what's going next. So finally, to wrap up, in terms of the
Ethan,
whole,
actually, quick question for you. Two questions for this one. There's muscle preservation, right? So BEMA last year showed some data. Looks like have some kind of a muscle preservation capability. But the one question a lot of people ask is, are those preserved muscle functional? Right? Yes, you have a mass. Yes, your muscle preserved, but do they really work? Right? That's always the question. Is number one. Number two, for corporate development pipeline management perspective, Regeneron is a tirzepatide kind of a fast follow from China, from Hansoh, right? So what's their plan? Obviously, semaglutide, glutide as a mono agonist probably was not enough. Do they plan to combine their dual agonist with their muscle preservation, give it one more shot? So two questions for you.
That's a very good question. So for the second question, we don't have answer. Regeneron usually is not very open on those kind of what's the plan next. You can talk to those people, but usually they're not that much transparent. So we don't know whether they're going to combine with tirzepatide. But you also notice that bimagrumab, the problem with bimagrumab is they increase the LDL cholesterol. So that's a major worry. So Eli Lilly, if you try to talk to Eli people, they don't know what to do with Bima. So they will probably wait and see. So you see, hitting the receptor don't have the nose bleeding issue, but they have the LDL increase issue. Then for obesity, any kind of safety signal is not welcome. So that's why Regeneron right now definitely they won't seeking activin A part. But myostatin, they are in a very tricky spot, is that you may need to design the trial long enough for the muscle to build. Then, yeah, I need to add some muscle strength test. So you can change the endpoints, the grip strength type of endpoint for phase 3 pivotal trial. You definitely need to show that your muscle is useful muscle, not just the lean mass on a DEX scan. So that's a very good point. But now their main hurdle is how do you convince the agency to modify that guidance, because it's still draft guidance. Hopefully, it can give you some leeway in terms of the balance between the muscle gain and the total weight loss. But it's an uphill battle, I have to say. There's no sugarcoating that. It's in a very tricky spot. So that's why in the final slide I have is a summary. So I don't need to repeat the first three. It's regardless of the low lean mass, but the final point you can tell Regeneron's kind of thinking. Additional research is needed to characterize the long-term clinical benefit and functional implication. So that's why they don't know what to do with the GLP-1. And the same thing happens to BIMA. BIMA even combined with GLP-1. Actually, once you stop BIMA, and muscle loss can be even worse and go below the baseline. That's another BIMA's dilemma, is that once you stop the drug, it can be worse than baseline after a certain number of weeks. And whereas GLP-1 and GIP, once they regain some weight, but they also regain some muscle. So that's why it's really a mixed bag in terms of we don't know the future about the muscle preservation. And that's why another hot topic I don't have time to cover is ALK7. I know so many companies are working on ALK7, which is a receptor for hepatokine. That may also give you some muscle preservation. So that's why the TGF beta family remain very fascinating. That's the key takeaway from the ADA conference. Yeah, Leon, back to you.
Yeah, thank you very much, Ethan. So I'm going to be really quick, just a kind of an open discussion, tidbits from ASCO and ADA. Each conference pick one talk point. I really want to invite everyone now. Let's just tidy up everything. This is not really about data. It's about like a vibe, right? The sentiment. So I'll start with ASCO. Actually, my good friends and my fellow newsletter writer, Angus, put this article there. Feel free to read it. I think it's fascinating. It's a kind of overview of the China Biotech presentation and ASCO. I mean, today, both Viraj and David already show a lot of presentation for the China Biotech, including the one of the two hottest topic, PD-1 VEGF, right? It's just influx of PD-1 VEGF molecule from China. Now they are presenting data. Sounds like my senses today, actually, people believe this is real. I mean, there's many others, right? Like a SAC TMT from ASCO, sorry, from Merck, even that's China-owned data. But based on my body language reading, David, you believe in their data? You believe their data probably going to translate into the global trial, and obviously the ADC and the PD-1 VEGF combo probably their hottest topic in oncology today. And then you're talking about in vivo CAR T. Wow, don't forget right after ASCO, Legend Biotech dropped their kind of EHA, which is gonna present their CD19, CD20 dual in vivo CAR T data over there, which actually add almost like $2.5 billion market cap for Legend at some point. And there's many, many others. So I think the vibe for China Biotech, regardless of how much the US want to resist them, their rise, they're going to continue to expand with or without the support of a US investor, early stage biotech or like a big pharma. So that's the first vibe. Second one, I'm going to finish real quick. There's something actually Ethan, gave me the tip, right? The speaker were taken out from the conference. Yeah, that was fascinating. So I'll pause here. Actually, someone waiting outside for me. So if you guys can share some quick tidbits, your reaction to this two headline, that would be great.
Maybe just to jump in really quick, I completely agree with you, Leon, in terms of the China, biotech being sort of like a almost like an incubator or breeding ground for like interesting next generation molecules, and we see that for VEGF PD-1, of course, where most of the pharmas actually license VEGF PD-1s, from China, and then also ADCs, where we're seeing quite a bit of good data from China, like Coolance, TMT of Merck, yes, Merck, and so there just tells you that there's quite a bit of innovation happening in China, and I don't think that's going to stop. I feel like there's quite a bit of negativity around the Trump administration saying that there's going to be like a ban on the sort of like the cross-border transactions, cross-border collaborations. But I feel like in some ways, yes, but based on my conversation with a lot of pharma, BDs, strategics, it seems like they're still looking, right? So it's not going to stop them until we see some additional detail around, what exactly is the government doing to like stop, China, or in licensing deals from China. So I don't think it's going to it's going to headwind for the time being. I think they'll continue to get more more assets, more interesting emerging technologies from China, especially if they're showing differentiated assets, differentiated data set. So yeah, so I would say definitely China's definitely a breeding ground for next generation oncology and I products going forward.
Yeah, I agree completely with David. I'm just gonna add one comment. So what this has done also is if you look at the last 5 years on what the follow-on time is from first in class to the next is significantly gone down. It was initially probably 5, 7 years or 10 years even at some point, and you had time to entrench yourself as as first in class. But now because of all this great innovation coming out of China, I think the follow-on molecules come out much quicker, and I think that basically underscores how you really closely need to think about what you're developing, where you're developing, and how what kind of unmet need you're addressing, and how you need to optimize clinical development. Because or basically if you think that you can remain first in class and remain a market leader in this space without thinking about what's coming out of China and what is because there are two things. One, it allows you to optimize quicker because you get all these different novel next gen agents that you want to combine with, you want to partner with, and which basically essentially accelerates all this great progress that we're making towards making cancer a potentially manageable disease, especially metastatic. So I think China innovation has contributed greatly to the fact that all of that optimized clinical development and combination strategy has been is so much front and center for all the companies, biotech and pharma.
Yeah, I would just simply add that during the ADA, you can see the Chinese companies, biotechs eager to present the data, but some of the US biotech now become very sensitive when they worry about disclosing things. If you search the name in the poster, they don't have any poster at the ADA. That means they don't want to disclose their target for fear of competition. That's why I want to wrap up. Yeah.
It's fascinating, right? So the kind of a dynamic change, but I guess that's a new reality. Well, get us kind of to be adaptive as adaptive as possible. That's just like the new business model now. All right. With that being said, thank you guys very much for taking the time. And I see there's some questions, especially people really like what we put out there. Obviously we are neutral. We don't want to take any political position, but that's just like a part of business today. Our audience is heavily focused on investment business, but we cannot ignore this not happening. This US-China, I call them US-China competition. There's a competition and a collaboration together. It's just a part of our business. With that being said, I really, really thank you guys very much for taking the time. I said there's a few questions. So let me say, what is that? Oh, there's just a quick comments from Charlene Liao. He really thank us for read this. Because of time, we cannot take the voice question anymore. If you do have a question for the speaker, please send the question to me. I will follow up. In the meantime, I really want to give a shout out our new Substack channel. So if you have any questions about the how to get a presentation, scan the QR code on the screen. Subscribe to our Substack account, and if you become paid member, well, you will have access to the presentation. So please support us. We are nonprofit, so we really, really appreciate your support. And then also leave a shout out to another episode in our pipeline. There's one in July 1st, actually already scheduled it. There's two more. And if you want to support us, please let us know. Finally, but definitely not the least, I want to give a quick teaser of our incoming July 1st webinar, really focused on ADC, really focused on, obviously, as you can tell today, that's where in oncology, the China biotech is really, really strong, right? So we actually want to bring this global perspective. We have a speaker from one of the largest pharma, Merck. We have actually the executive from South Korea and the US biotech company. We have a bunch of the leading ADC company based in China all together, and also big pharma moderator from EMD Sloan, actually European big pharma together to talk about ADC. Can be fascinating. And please register, and we'll see you in 3 weeks. With that being said, thank you, David. Thank you, Viraj, and thank you, Ethan, for taking the time. And thank you, everyone. Stay with us until the very end. We run it over time, but I think it was totally worth it. With that being said, thank you very much, and see you next time. Thank you.
Thank you,
Thanks,
Leon. Thank you,
Leon. Thanks,
everyone.
Thank you.
Thank you.
Bye-bye.
Bye.
Bye.